Hello, everyone. Welcome to part 2 of chapter 3. So, in this chapter, we're going to actually finish off the chapter. We'll be focusing a lot on other things going on with the cell now. We've done the cell membrane. We discussed all the way up to organelles and gene expression, basically. So now what we're going to do is we're going to talk a little bit more about the cell membrane, talk about how we move through the cell membrane, talk about that a little bit. So what we want to do is talk about how, remember that it's a barrier. But the thing is, it's not a perfect barrier. It's not completely a barrier that nothing can get through. It is. So one of the things I like to basically say to my students is this. Your cell is not self-sufficient. What I mean by that is cell must obtain nutrients. It must get rid of waste. Okay? It's got to get things in. It's got to get things out. It's not completely self-sufficient. One of the ways that I can say that is in my biology classes, I like to remind them and talk about how the cell is what we would say is an open system in thermodynamics. Means it is open to matter and energy. So that means stuff needs to come in and stuff needs to leave. So now it's going to be this idea of permeability that determines what can get in or get out. Now, a cell could be impermeable. It could be nothing gets in or gets out. It could be freely permeable, and that could be anything gets in or gets out. Well, cell membranes happen to be selectively permeable, and that means what can get in gets in, what can get out gets out. Okay, it's selective. Now, that means if I look at a kidney cell will be permeable to certain things and impermeable to others, where a neuron would be permeable to other things and impermeable to others, and etc. So what we'll see is cells have this selective permeability that they get to be permeable to. Okay, they are selectively permeable. Oh, what gets in, what gets out, gets out. Okay. Okay. Now, how is this selective permeability determined? Well, there are a few big things. There are 4 big things, and some of these are tied together. But I always like to talk about this, and basically it's a rule of thumb that I always like to say is the 2 biggest factors that will influence this, that I always tell my students, is small and nonpolar. Okay? Small and nonpolar. So kind of keep that in mind. Small, nonpolar, the easy. Okay? Now, so size. The smaller the molecule, the easier it diffuses. Electrically charged, nonpolar molecules diffuse better. Some molecules, if they're charged, they can't. They have to go through a protein. If it's bulky, it doesn't fit. If it's lipid solubility, if the cell is mostly lipids, cell is mostly made up of lipids. So only lipid soluble molecules go through the cell membrane. So lipid soluble molecules will diffuse better, more easily. Okay. So what we want to do is discuss this idea that if you're small and nonpolar, you go through very easily. And that will be a good kind of framework to remember. Okay, that is the statement, small, nonpolar, passes through cell membrane, easy, easy, easy. And that will be the big thing to remember. Now, when you do cross that, you can cross actively or passively. Passively means I pass the cell membrane with no energy required. Actively means I do require energy, and that's usually going to be ATP that the energy is going to be provided by, usually an ATP. Now, let's talk about one way, a type of passive movement that involves molecules moving, and that is high to low, and that is diffusion. Diffusion, I frequently like to say diffusion is like a fart, okay? Well, it goes from high to low. If you break wind in the front of the classroom, and that diffuses, it will spread out from a high concentration to a low concentration, okay? And eventually it will spread out throughout the room, and eventually it becomes diluted and no one can smell it, okay? Diffusion is the movement passively of materials from a high concentration to an area of low concentration, okay? Now, anytime we have a system, that there is a high concentration on one side and a low concentration on the other side, we call this a concentration gradient. What we say is diffusion follows the gradient. It goes from high to low. Now, and another important thing is there's this idea we talked about, equilibrium is when there's equal concentration through the entire system. Right here, we are in equilibrium. Once we're in equilibrium, there's no more high to low to go. So when it goes from high to low and it reaches equilibrium, it must stop. Okay? Now, that's when it stops. Now, diffusion is very important because this is how we're going to move carbon dioxide and oxygen and things like that from cells to blood and blood to cells and from cells to cells is diffusion. We're going to move things a lot of times through diffusion. Diffusion is very important in physiology. This is how we're going to move ions in neurons. This is how we're going to move ions in a lot of the kidney stuff. This is how we're going to move stuff in the body is diffusion. Now, this diffusion we'll talk about is affected by a variety of things. Diffusion is affected by a variety of factors that can influence how fast. Now, it does matter. Now, distance, this is a very important one in biology. In any biology class, the distance is very important. Now, why is it important is diffusion is like a sprinter. It is a short distance runner. It does not do long distance. The shorter the distance, the more quickly, okay? Short distance diffusion. It likes short distance. Molecular size. Small things have faster diffusion. So you move more faster if you are going a short distance. More quickly, concentration gradients are eliminated. This should say gradient right here, not concentration grades, a concentration gradient. I apologize. Sorry, I didn't enable editing. This should say gradient, not grades, gradient. Let me highlight that, make it red for you guys, red and highlighted, just so that stands out my typo there. Concentration gradients get eliminated, okay? Now, molecular size, smaller things, they're going to be faster. Temperature, higher temperature make things go faster, okay? Concentration gradients, the steeper the gradient, the faster diffusion moves. And then another thing is electrical forces. Opposite charges will attract each other and like charges will repel. Okay, so they will repel each other. Okay, so so like charges plus and plus, negative and negative. Okay, all right. Now, when you diffuse, you can go by what we call simple diffusion or facilitated diffusion. Simple diffusion is simply going through the phospholipid bilayer. This is things like fat, steroids, oxygen, carbon dioxide, a lipid-soluble drug, things like that. These are all that diffuse through the lipid part of the cell membrane, the lipid portion of the cell membrane. Facilitated diffusion, on the other hand, this is going through a channel where the channel protein, a protein facilitates the movement. So right here we have a steroid, 4 fused rings. This is cholesterol. This is the structure of cholesterol. This cholesterol simply goes through the phospholipid bilayer because it is hydrophobic. Okay? Now, facilitated diffusion. If you are hydrophilic, like this substance right here, who is working on an ion, is working as an ion probably, or a very small nonpolar substance, how do we know it's nonpolar? Is this water is interacting with it. So it's dissolved in water and it goes through a protein. That's called facilitated diffusion. This allows non-lipid soluble things, hydrophilic compounds, things like that, ions, glucose, water, that kind of stuff, to pass through a protein. Okay? It goes through a protein to get through the phospholipid bilayer. All right, now here's a big one, osmosis. Osmosis is a big one. Osmosis is diffusion of water. Water, the diffusion of water, that means water goes from high water to low water. Now, this is where we get into something kind of weird. How do we have high water? How can we have high water? Let's help you understand this, okay? Let's try to understand how we can have high water. Now, let's say I have a water molecule right here. So this is a water molecule. There's the oxygen, partial negative, okay? Here's the hydrogen, partial positive, okay? Now, let's just say that I take NaCl and I add it to water. I put it in water. This will ionize or dissociate and form Na plus and Cl minus. Now what's going to happen here is, guys, this Na plus is going to interact with the opposite partial negative of water, and the Cl minus is going to interact here. Now what's going to happen is we talked about water as a solvent, that water does this thing where it surrounds stuff that it dissolves, okay, and formed a hydration sphere, okay? We remember that from chapter 2, okay? It forms a hydration sphere, okay? So it's going to do that over here. Now remember, what is happening here is these water molecules, when you dissolve things, water molecules are being spent, being expended to keep them dissolved and dissolve them. So these hydration shells that are forming here, this is like water molecules being used up to keep these things dissolved. So anytime that ever I increase a solute, this will mean that I decrease the amount of water. And anytime that I lower a solute, this will mean that I will increase the amount of water. And right here is the reason why. Solutes are going to expend water, keep water tied up, so to speak, to dissolve it. Okay? Makes sense? This is how we can have high water and low water. Okay. So now that we got that out of the way, and I hope you understand that, we have this idea that water diffuses, moves from high water to low water. Now what we can basically say is this. Osmosis, it's a real hoot. Water always moves towards a greater solute. Okay? That is the simplest way that I could ever explain osmosis to you. Okay? Now, osmolarity. That is the number of particles dissolved in water. That's how many particles we got. Tonicity is the effect that that has on the ability of a cell to lose or gain water. Osmotic effect of a solute on the cell. Now, you can have two fluids have the same osmolarity, but different tonicities. Why? They can have the same number of particles dissolved in them, but they're not necessarily the same amount of the same particles. You can have different amounts of sodium and different amounts of potassium, but have the same amount of total particles dissolved, but not be the same number of sodium and the same number of potassium. So when we compare that, they would have different tonicities, but the same osmolarity because they have the same number of particles. Osmosis, it's a real hoot. Water always moves to the greater solute. Now, why is that? Why does water move to the greater solute? The solute is hydrophilic. It's hydrophilic. Water likes it. It likes water. Water will attract to that stuff. It will attract water to itself. Water will get attracted to it because it is polar. So what will happen is when water is attracted to it, the force that a pure water or water that is low solute, so this means water will move. Water moves towards a greater solute. This is called osmotic pressure, the force generated when pure water moves into an area of higher solute concentration. We would say that a low solute liquid has a higher osmotic pressure than a high solute liquid. Okay? And this is a type of hydrostatic pressure, the force generated of a fluid pushing against a fluid. Force generated from pushing against a fluid. So when you push against fluid, you have hydrostatic pressure. Okay? So, all right. So osmosis, it's a real hoot. Water always moves to the greater solute. Okay. Now, let's explain this, okay? Osmosis. Let's say I have some beakers. Okay. And in these beakers, I have some water. And in this water, I place a little red blood cell. So we have a cell in here. Now, this cell is floating around in this water. Now, this cell has got some solutes in it. Let's say it's got these solutes, 3 solutes in it. And the cell is the same because cells maintain homeostasis, basic cell theory. Okay, now, the world around it can be the same. We would call that isotonic. Iso meaning equal, tonos meaning pressure. They have the same osmotic pressure. Water moves from high osmotic pressure to low osmotic pressure. It's one of the ways we can say this statement, okay? Water moves towards the greater solute. Now, water moves from high osmotic pressure to low osmotic pressure. Now, here's the thing. There's no greater osmotic pressure. They are the same. So what water will do is water will move in and out equally. H2O. This will be H2O. I couldn't write it any finer than that because I don't have a fine point marker. Water will move in and out equally. Okay. Now, I believe in my notes that I have hypo next. Okay. Now, say we only have one here. Osmosis, it's a real hoot. Water always moves towards the greater solute. Water will go in. What this cell will do, when water goes in, this cell will swell up. The cell will swell. This environment that we're in that is low is hypotonic. The solution has a low osmotic pressure. Water moves from high osmotic pressure to low osmotic pressure. So osmosis, it's a real hoot. Water always moves towards the greater solute. So let's say we had 1, 2, 3, 4, 5, 6. We had a really high solute concentration here. This solution here is called hyper, which means high tonic pressure. Hyper, high tonic pressure. There is a high osmotic pressure outside, and water goes from high to low osmotic pressure. So this hypertonic here, this actually has the higher osmotic pressure. Sorry, this has a lower. It means so this cell here. What's going to happen is, guys. Sorry, I messed up for a second. I don't want to restart the recording. I'm tired. Sorry, I had a big bunch of classes today. Sorry about that. So it means high pressures. The cell here, okay. So the cell is going to have a higher osmotic pressure than the stuff outside. So what's going to happen is it's going to draw the liquid out. And I apologize. I about said something wrong there, and I didn't want to say something wrong, but I'm pretty tired. Okay, there we go. Water's going to move out. Okay, water's going to move in. Water's going to move out. What this cell is going to do is it's going to be able to shrink. It's going to want to move in. It's going to shrivel. It's going to shrivel up. Okay, shrivel. Now, Sorry, I meant to go here. Sorry, I don't want to start over, but I'm not going to start over because, gosh, I already said way too much stuff that I don't want to restart myself. I'm sorry if I. So isotonic, it does not cause osmotic flow because the concentrations are equal to each other. Okay, equal in concentration on both sides. Since the ECF and the ICF are equal, there's no net water movement. Hypotonic causes flow into the cell. Low concentration of solutes in the ECF. This causes swelling, maybe bursting. We call this hemolysis. Hypertonic causes water to move out. High concentration of solutes in the ECF draws water out, results in shriveling. We call this crenation. Okay, cells crenate. And this is what's happening here. Okay. Okay. Sorry about that one. My brain got a little bit. I'm a little bit mentally tired, so I apologize if I made a slight weird statement there. I hope I did not confuse you. Okay, carrier proteins. These are integral proteins. They carry ions across the phospholipid bilayer. They can carry substances. Sometimes they carry one substance. Sometimes they carry two. Sometimes they carry more. When it comes to two or more, there's one called cotransport or symport. That's two substances in the same direction at the same time. And countertransport or antiport, that is two substances across the cell membrane in opposite directions at the same time. Okay, counter to each other. I'm going to give you an example of those, but know those because these are very important for drugs. We give drugs that target symporters, antiporters, things like that. Now, passive transport or facilitated diffusion requires going through a protein. They transport hydrophilic substances like glucose or ions, and what these things have to do is change their shape. For example, there's a protein that transports glucose that does this flip-flop shape change. There's various glucose transporter classes, like a glucose transporter class 2. It changes its shape, GLUT2s. They don't require energy, but they just follow up. They require a concentration gradient, a high to low. Now, active transport, this is a very big deal. Active transport is transporting material through the cell membrane using energy, usually ATP. Now, one example of active transport where ATP is not used would be in the electron transport chain of mitochondria, or in bacteria where the electron transport chains are used to generate chemiosmosis. Okay, now this can go against the gradient, high to low, low to high. Okay, it can go from low to high, actually, against the gradient. It can go from a low concentration to high and make it even higher. Primary active transport is the active transport that actually uses the energy. There's one called secondary that doesn't. We're going to learn about that. So let's do a primary active transporter, and I'll come back and draw this out, and then we'll talk about it. Okay, now, here we have a cell, and we have a fluid. We have our ICF, and we have our ECF. Now, how are we going to get these? We have a protein. Now, let's just say, for example, I have this protein right here, and we have a protein embedded in the cell membrane. And let's say this protein, what it does is this protein will take in ATP, okay? We know that's the energy of a cell, ATP. And it takes ATP in, okay? ATP comes into it, binds to it. And this ATP allows it to do something. And one of the things it allows it to do is to pump in so much stuff and pump out so much stuff. And it might pump out 3 sodium cations and pump in 2 potassium cations. Okay? For every ATP we give it. Now, this would be the sodium-potassium pump. 2 potassium cations in, 3 sodium cations out. 3 sodium cations out, 2 potassium cations in. Now, it's actually more accurate to say 3 sodium cations out, 2 potassium cations in, because that's the order of the pump. But know it forwards and backwards, okay? Know it which way. The sodium potassium pump. Sodium potassium ATPase pump. So this is the sodium Na plus / K plus ATPase pump. Sodium potassium pump. Sodium potassium ATPase pump, okay? Now, let me give it, this is a primary active transport protein, okay? And it's the most important. But let's go a little bit further. This is how it does. 2 potassiums in, 3 sodiums out. 3 sodiums out, 2 potassiums in, okay? For every molecule of ATP that I give it. Good, 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 good. Okay, so let's look at this ECF. This ECF happens to have a fair amount here in the ICF. Sorry, I was pointing towards the inside the cell. The ICF. The ICF tends to have a high potassium and a low sodium content. And the ECF will have a high sodium, low potassium. And we might basically say this cell right here is going to be like a salty banana. Why is it like a salty banana? Because it's got a lot of sodium outside and a lot of potassium inside. Salty banana. Imagine a banana rolled in salt. Okay? Now, because there is an unbalanced charge here, 3 sodiums out and 2 potassiums in, this creates an unbalanced, unstable charge. The ICF will be quite a bit more negative, and the ECF will be quite a bit more positive, and we refer to this as being a polar charge. Okay? All right. So the sodium potassium ATPase pump, the most important primary active transporter in the body. 3 sodiums out, 2 potassiums in. 2 potassiums in, 3 sodiums out. However you want to do it. It is so it pumps out that particular set of things that it pumps. Now, it results in the ICF having high potassium, low sodium, and the ECF having high sodium, low potassium. ECF will be slightly more positive because it has more cations, and the ICF will be slightly more negative charge because it has less cations, and you will have an unbalanced, unstable polar charge. Okay? Now, secondary active transport. Let's explain that. Let's say I had a transporter over here, okay? And this transporter that I've got right here, this transporter is a glucose-sodium cotransporter. Now, let's say I want to bring glucose into a cell, and because cells need glucose for cellular respiration. So I want to bring some glucose into this cell, and that's a very important thing for a cell to get its hands on is glucose. And I need to get some glucose into this cell. Well, as it turns out, sodium was pumped out to high concentration. So what if I took this sodium like this that I pumped out? Let's do it this way. I could write better than that. Come on. What if I did that? And with that coming in, I brought glucose in too. This is primary. This is secondary. This uses ATP, sets the gradient to bring glucose with it. This kind of works like, hey, you're going to the mall. How about I ride with you? And how about my friend rides with us since you're already going to the mall? You're already driving to the mall. Let me ride with you. And won't you take my friend with us too? Okay, that's how secondary works. Okay, hope that made sense. Okay. So basically, secondary active transport is driven by the primary active transport. Primary active transport drives it. It does not require energy, secondary active transport. It uses the primary active transport to do all the work. Now, for example, the sodium potassium ATPase pump does all the concentration gradient setting. Then a second one can use that gradient. Like, for example, the sodium potassium pump does the work, and the glucose sodium transporter just piggybacks off of it. Sodium goes from high to low, and it brings glucose with it, regardless of the concentration of glucose. It just is one of those ways that we can bring glucose into a cell because cells need lots of glucose because cellular respiration. Okay. Now, there's another way we can bring things, and that's vascular transport. Vascular transport, so the means of transport, is a type of active transport. It's bulk movement of material into or out of a cell where a vesicle is used. It's always active because ATP is required to move vesicles on microtubules. Excuse me, got the hiccups. And we have an endocytosis. That's transport into a cell using a vesicle. And we have exocytosis. That is transport out of a cell using a vesicle. Okay. Now, for endocytosis, there's three kinds. There's receptor-mediated endocytosis, there's pinocytosis, and there's phagocytosis. Now, the vesicles of endocytosis, we call this an endosome. If it's for receptor-mediated endocytosis, we call it a coated vesicle. If it's pinocytosis, we call it a pinosome. If it's phagocytosis, we call it an endosome. Okay? So let's look at these in more detail. Receptor-mediated endocytosis is very selective. What we have here is we happen to have these receptors that are bound to the outside of the cell. They're bound to the outside of a cell. And we have these molecules, these target molecules called ligands, and they're floating around outside the cell. They bind to these receptors, and what they're going to do is when enough of them bind, we fold these in and form an endocytosis and form an endosome called a coated vesicle. And then what we can do is we can get these molecules off and remove them and use them, and maybe they're things we need in a large amount. So that's how we can get large amounts of certain specific targets in. These target molecules are called ligands. Okay? Now, vascular transport, pinocytosis and phagocytosis. Pinocytosis is called cell drinking. Think about this. You might drink a wine called a Pinot Noir or a Pinot Grigio, a Pinot wine, Pinot. Pinocytosis means cell drinking. It is the bulk transport of extracellular fluid, ECF, into a cell. Phagocytosis is a cell eating. This is cells bringing in bulk stuff from the outside world, solid materials. It might be a bacterial cell, for example. A bacterial cell gets phagocytized. We take a lysosome to it. We bind to it. We gobble it up. We chew it up into little pieces, and then we spit it out. Sometimes before we spit it out, we find one of these pieces, and we make an antibody. We grab it, and we make antibodies against it. We end up making antibodies against those little pieces, and that's how we can fight bacterial infections. The endosome is called a phagosome. Endocytosis, this is, that's the ones we learned. Now we got exocytosis. So exocytosis is transport out of a cell using a vesicle. Here we have a vesicle. It comes and binds and fuses to the cell membrane, and because they're made of the same stuff, they're both composed of phospholipids. They fuse together. They're both made of phospholipids. They fuse, and we release the contents. This is exocytosis, is where vesicles are removed out of the cell. Things are removed by a cell. Exo, out of. Cytosis, cell. Transport out of a cell using a vesicle. Exocytosis. All right, that's the transport mechanisms. All right, so diffusion, simple diffusion, simply through the phospholipid bilayer. Facilitated diffusion through a protein. Active transport needs ATP to make it happen. Okay. One of the last things we're going to talk about today is the cell cycle. Okay. Now, the cell cycle is one of the last things we want to talk about. And remember, cell theory says all cells come from other cells. If all cells come from other cells, we got to take a cell that existed and divide it to make new cells. This requires cell division, where a single cell divides and becomes daughter cells. Now, cells do have a lifespan. We were just talking today in Biology 2, A&P 2 actually, about how long red blood cells last and what happens to them when they die. Cells, sometimes they will self-destruct. Many cells, when they reach the end of their lifespan, self-destruct in a programmed cell death. We call this apoptosis. And now cell division does require me to duplicate my DNA. That is a requirement for that. Now, nuclear DNA is wound up into chromosomes. We have two sets. Normal human cell has 46 chromosomes in it. Now, these chromosomes coil up and they have these two things called chromatids. We call these sister chromatids. Now, here's the deal. Sister chromatids are identical. Now, the deal is, your sister chromatids are identical. Your sister probably is not identical to you. If you have a sister, your sister probably is not identical to you, but if you sister chromatids, they are identical. Now, the two sister chromatids are on the same chromosome. They are genetically identical. They are the product of DNA replication. If you took biology, you know how DNA replication happens, and we know that basically that happens during S subphase of mitosis and all that. Now, they are genetically the same, okay? Now, chromatids connect to each other at a centromere. The centromere has these little binding spots called the kinetochores. The kinetochores are going to be like trailer hitches to bind to the chromosome, where you're going to take your microtubules and bind to them and yank them apart and pull them around like a trailer hitch. Okay? Now, to do this, we need cell division. Cell division takes a single cell and divides it into two daughter cells. Those daughter cells will be half the size of the original cell. Now, to do this, we must first take our DNA and replicate it. It must be replicated. It's called DNA replication. Then we go through a process of division called mitosis. This division, if it happens in somatic cells, is called mitosis. If it happens in gametes, we call it meiosis. We will discuss this more AP2. But here's what I want you guys to know. In mitosis, we make somatic cells. In meiosis, we make sex cells. That's gametes. Mitosis always makes exact copies. Meiosis never makes exact copies. It makes non-identical copies. Every one is a uniquely different cell. In mitosis, all cells possess two copies of each chromosome. We call those cells diploid. That is a fancy word. If you have two copies of each chromosome, you're called diploid. All cells possess only one copy of each chromosome is haploid. Okay? Haploid is one copy of each chromosome. If a cell begins with 46 chromosomes in mitosis, and mitosis happens, there will be 46 chromosomes in the daughter cells. But in meiosis, if you begin with 46 chromosomes and undergo meiosis, you'll have 23. Always half. Okay? Now, if we were to look at a clock, most of the clock would be devoted to interphase. Interphase is a good chunk of the cell's life. It is called interphase. Interphase is where most of the life of the cell is spent, is in a non-dividing state called interphase. But interphase has got three small phases, and what it does is it gets ready to divide. Cells spend only a small part of their time actually dividing. Majority is an interphase where it's functional, but it gets ready to divide if needed. There is preparation for division if we need it. One of the things it does, we have G1, S, G2. Go, Sally, go. Go, Sally, go. That's interphase. Go, Sally, go. Growth, G1 is growth and duplication of organelles. Then S is DNA synthesis or replication. And G2 is protein synthesis phase. Okay. Now, M phase, mitosis, is when we divide the genetic material. Think of interphase as preparing for a breakup. Think of mitosis as the actual breakup. Think of cytokinesis as when you say goodbye to each other. Okay? Interphase, you know you're getting ready to break up with somebody. You see the signs of breakup is coming. Mitosis, you're like, here's your ring back. Here's your hoodie back. Here's this thing you gave me as a present. I don't want it anymore because we're breaking up. Cytokinesis, well, I guess this is goodbye. So interphase, you are getting prepared to break up. Mitosis, you break up. Cytokinesis, you say goodbye to each other. Okay? Go, Sally, go make children. Cytokinesis is when you actively divide the organelles into two genetically identical daughter cells and they're ready to go. Go, Sally, go make children. Okay? Now, one of the things I will test you guys on is what happens during mitosis. Now, what we're going to do is we're going to start with prophase, sometimes called early prophase, and late prophase, prometaphase. There's prophase, the first phase, before phase, the phase that's before. So prophase, before phase, it's the phase that begins before. It begins with chromatin condensing and chromosomes being visible. Your spindle fibers will be visible. The astral rays are visible and radiating. Then in prometaphase or late metaphase, you have two copies of each chromosome now exist. The nuclear envelope breaks down. You have what's called a chromatid gets bound to a centromere. The chromatid as a centromere, it gets bound the kinetochores to a microfiber, to a spindle fiber, microtubule, chromosomal microtubules. Okay, and that's when you're going to grab a hold. So your nuclear envelope breaks down, then you grab a hold of the chromosomes. Metaphase, you move them in the middle. They move to a narrow central zone called a metaphase plate. Meet in the middle. Once they're aligned at the metaphase plate, we end metaphase. Anaphase, you're going to separate the sister chromatids. Sister chromatids separate. The chromatids split, and the chromatids separate, and they're pulled towards the opposite poles. Then in telophase, they are going to be put into opposite cells. The nucleus reforms. The nuclei is there. Chromosomes uncoil. That's the end of mitosis. Then the cells separate, and that's cytokinesis. Now this will begin what we have called a cleavage furrow to be completed and be continued from telophase. Telophase has a structure called a cleavage furrow that starts to form, and cytokinesis, when it's completion, that's the end of cell division totally. So what we will see here is, guys, is that we have, first off, we take our little centrioles, our pairs of centrioles, okay, our centrosomes, and they start producing your little rays, and they make astral rays. Nuclear envelope is here, but then we will start to see chromosomes start to form. Chromosomes will start to form. Then the nucleus breaks down. Those astral rays, they grab a hold of the chromosome, about the kinetochore. They grab them by the kinetochore. Then what they do is they line up during metaphase and the metaphase plate. Then during anaphase, they separate. Then during telophase, a cleavage furrow will start to form, and they will be separated into nuclei, and then cytokinesis, they will completely separate. So one of the things I will ask you in a test is what happens during this, okay? Now, there are a couple things I want to talk about, and that is cancer. Cancer tumors, called a neoplasm, is a mass that is formed by abnormal cell growth and division. Cell division that we just talked about is an important part of cancer. If a tumor is benign, it's contained, it does not spread, it is contained. It's not life-threatening unless it's large enough to cause a problem. Malignant tumors, however, spread and invade surrounding tissues. We call that metastasis. Cancer is the result from abnormal proliferation. There's mutations in the genes that regulate growth. These modified genes, we call them oncogenes. An oncogene is any gene that has the potential to cause cancer. In tumor cells, they're mutated or they're really turned on too high. How that can happen is mutagens. They can be any agent that causes mutation. And there's carcinogens. Those are mutagens that are cancer-causing agents. Things like benzene, asbestos, and that kind of stuff. Metastasis is when we spread to other areas. We invade the tissue and it spreads. Okay. So an abnormal cell may start off growing in a place, grow into a tumor, then it may invade and metastasize and set up somewhere else. Okay? All right, we're done. So if you guys guess that this is, go ahead and guess what these are, and then pause and see if you got the right answer. This is an isotonic, hypotonic, hypertonic. All right. Guys, this concludes that. It means you've got a quiz to do, so get ready, and I'll see you in the next one.